day everyone, I am Maria Isabel Argonar from BSL 3 Science, and I am here to share to you the characteristics of some parasites. And these parasites are namely Naegleria fowleri, Necator americanus, and the Paragonimus westermani. But before we proceed to that discussion, we will be having an activity entitled I Want to Go Home. So here are the directions. So okay, first let us discuss about the Naegleria fowleri. So this Naegleria fowleri is a free-living amoeba with a transient flagellate stage and resistances in its life cycle. So one species, Naegleria fowleri, causes primary amoebic meningoencephalitis or the PAM in humans. They were subsequently maintained as clonal cultures derived from single cells together with the local clinical isolate that was previously designated as M. It is belong to the kingdom of Protozoa, phylum of Protozoa, class of Lobosa, order of Amoebida, family of Balcampidae, genus of Naegleria, and species of N. fowleri. Okay, now let's proceed to the life cycle of this Naegleria fowleri. So Naegleria fowleri has 3 stages in its life cycle. So we have the cyst, the tropozoid, and the flagellate. So the only infective stage of the amoeba is the tropozoid. The tropozoids are 10 to 35 micrometer long with a granular appearance and a single nucleus. The tropozoids replicate by binary division, during which the nuclear membrane remains intact. So this process is called promitosis. So the tropozoids will infect humans or animals by penetrating the nasal tissue and migrating to the brain, to the olfactory nerves, causing primary amoebic meningoencephalitis or the PAM. Now let us discuss for its epidemiology. So invasive human infections have been reported in Australia, which is where this organism was first identified, and New Zealand, Europe, Africa, Asia, and Latin America. So in the United States, it has been isolated from the thermally polluted waters in the northern states like Connecticut and Minnesota. As well, there have been 34 cases reported in the United States from 2008 through 2017, and a total of 143 infections were reported. So how does these infections transmitted? So Naegleria fowleri infects people when water containing the amoeba enters the body through the nose. This typically occurs when people go swimming or diving in warm freshwater places like lakes, rivers, and this Naegleria fowleri amoeba then travels up to the nose to the brain, where it destroys the brain tissue. Okay, so now let us discuss the pathogenesis of this N. fowleri. So this N. fowleri is dependent upon the release of cytolytic molecules, including acid hydrolases, phospholipases, neuraminases, and the phospholytic enzymes that play a role in host cell and nerve destruction. So the combination of the pathogenicity of N. fowleri and the intense immune response resulting from its presence result in significant nerve damage and subsequent or CNS tissue damage, which often refers or results in death. Is N. fowleri. So despite the absence of the specific signs and symptoms of this infection, the most common symptoms include severe headache, fever, chills, positive Brudzinski sign, positive Koenig sign, photophobia, confusion, seizures, and a possible coma. In addition to these, cardiac rhythm abnormalities and myocardial necrosis have been observed in some cases. So okay, now let us proceed to the symptoms of this Naegleria fowleri. So this Naegleria fowleri is divided into two stages of symptoms. So for its stage 1, we have the severe frontal headache, fever, nausea, and vomiting. While in stage 2, we have the stiff neck, seizures, altered mental status. Next is the hallucinations and possible coma. So how can we prevent this kind of infections? So there are 3 given ways on how can we prevent it. So we have number 1, don't swim in or jump into warm fresh waters like lakes, rivers, like that. Then next is hold your nose shut or use nose clips when jumping or diving into warm bodies of fresh water. Next is avoid disturbing the sediment while swimming in shallow, warm, fresh water. Okay, so the question is there any treatment for this kind of infection? So yes, there are recommended treatment for Naegleria infection, and it is the combination of drugs including amphotericin. So this amphotericin B is antifungal drug that is usually injected into a vein or into the space around the spinal cord to kill the amoeba. So next is the miltefosine, which is an investigational drug that is usually used to treat breast cancer and leishmaniasis, but has also shown promise against the Naegleria amoeba in laboratory and animal studies. It has also been used to successfully treat infections with other types of amoeba. So the other recommended drugs are antifungal drugs and antibiotics. Okay, so here are the references. So now let us proceed to the next parasite, which is the Necator americanus. So for its morphology, this hookworm developmental stage including eggs, 4 larval stages, and adult worm. So eggs appearance or appear as oval, thin shelled bodies measuring 55 to 77 micrometer in length by 35 to 42 in width. Freshly excreted eggs contain a developing embryo in the early stage of cleavage, which is 2 to 8 cells. So okay, for the classifications of Necator americanus, so it is belong in the kingdom of Animalia, phylum of Nematoda, class of Chromarhodea, order of Rhabditidae, family of Ancylothamatidae, and the genus of Necator and species of Necator americanus. So now let us proceed to its life cycle. So eggs are passed in the stool and under favorable conditions like moisture, warmth, and shade. So larvae hatch in 1 to 2 days. The released rhabditiform larvae grow into the feces or in the soil. So and after 5 to 10 days, they become filariform in the 3rd stage, so which is the larvae that are infective. So these infective larvae can survive 3 to 4 weeks in favorable environmental conditions. So on contact with the human host, the larvae penetrate the skin and are carried through the blood vessels to the heart and to the lungs. So yes, they are also penetrate into the pulmonary alveoli, ascend the bronchial tree to the pharynx, and are swallowed. So the larvae reach the small intestine, where they reside and mature into adults. So adult worms live in the lumen of the small intestine, where they attach to the intestinal wall with resultant blood loss by the host. So most adult worms are eliminated in 1 to 2 years, but the longevity may reach several years. Okay, so now let us discuss its epidemiology. So hookworm thrives in regions extreme poverty with poor sanitation and affects some 740 people worldwide. So developing nations with warm, moist climates in regions like South America, South Asia, and Southeast Asia. These are most susceptible to the worm. So in the United States, it has been isolated from the thermally polluted waters in the northern states like Connecticut and the Minnesota as well. There have been 34 cases reported in the United States from 2008 to 2017, and a total of 143 infections were reported. So how does these infections transmitted? So we can get these hookworms by walking barefoot over contaminated soil, in penetrating your skin. So the hookworm larvae or the immature worms may cause an allergic reaction and are carried to your lungs. The larvae migrate from your lungs to your windpipe and are then swallowed and carried back down to your intestine. Okay, so now let us proceed to its pathogenesis. So the hookworm species that reach maturity in a human intestine have similar life cycles. So eggs pass in the stool, hatch in 1 to 2 days, and release rhabditiform larvae, which molt once to become slender fowleriform larvae in 5 to 10 days. So the larvae can survive 3 to 4 weeks if environmental conditions are favorable. So fowleriform larvae penetrate human skin when people walk barefoot or otherwise come into direct contact with infected soil. So the standard method for diagnosing the presence of hookworm is by identifying hookworm eggs in a stool sample using a microscope, because eggs may be difficult to find in light infections. A concentration procedure is recommended. So okay, for its symptoms, so ankylostomiasis is infection with the hookworm ankylostoma, and duodenal or Necator americanus symptoms include rash at the site of larval entry and sometimes abdominal pain or other gastrointestinal symptoms during early infection. So later, iron deficiency may develop because of chronic blood loss. So how to prevent this kind of infection? So the best way to avoid hookworm infection is not to walk barefoot on areas where hookworms are common and where there may be human fecal contamination of the soil. So also avoid other skin contact with such soil and avoid ingesting it. So okay, for the treatment for this infection, so there are anthelmintic medications or drug that rid the body of parasitic worms such as albendazole and mebendazole. So these are drugs of choice for treatment of hookworm infections, and these infections are generally treated 1 to 3 days. Okay, so here are the references. Okay, so now let us proceed to the last parasite, which is the Paragonimus westermani. So this Paragonimus westermani is an eggs range from 80 to 120 micrometer long by 45 to 70 micrometer wide. They are yellow-brown, ovoid or elongate with a thick shell and often asymmetrical with one end slightly flattened. So at the large end, the upper column is clearly visible. So the opposite ovipocular end is thickened. So the eggs are unembryonated when passed in sputum or feces. Okay, so for its classification, these Paragonimus westermani is belong to the kingdom of Animalia, phylum of Platyhelminthes, class of Rhabditophora, order of Flagellorchida, family of Phragiminidae, and the genus of Paragonimus, and the species of westermani. So okay, for its life cycle, the eggs are excreted unembryonated in the sputum, or alternately they are swallowed and passed with stool. In the external environment, the eggs become embryonated and myristidia hatch and seek the first intermediate host, a snail, and penetrate its soft tissues. Myristidia go through several developmental stages inside the snail: sporocysts, radiae, with the latter giving rise to many cercariae, which emerge from the snail. The cercariae invade the second intermediate host, a crustacean such as crab or crayfish, where they exist and become metacercariae. This is the infective stage for the mammalian host. Human infection with P. westermani occurs by eating inadequately cooked or pickled crab or crayfish that harbor metacercariae of the parasite. The metacercariae exist in the duodenum, penetrate through the intestinal wall into the peritoneal cavity, then through the abdominal wall and diaphragm into the lungs, where they become encapsulated and develop into adults. The worms can also reach other organs and tissues such as the brain and striated muscles, respectively. However, when this takes place, completion of the life cycle is not achieved because the eggs laid cannot exceed these sites. Time from infection to oviposition is 65 to 90 days. Okay, so now let us proceed to its epidemiology. So several species of Paragonimus cause most infections. The most important is P. westermani, which occurs primarily in Asia, including China, the Philippines, Japan, Vietnam, South Korea, Taiwan, and Thailand. So P. africanus causes infections in Africa and P. mexicanus in Central and South America. So especially dishes in which shellfish are consumed raw or prepared only on vinegar, is brine or wine without cooking, play a key role in the transmission of paragonimiasis. So raw crabs or crayfish are also used in traditional medicine practices in Korea, Japan, and some parts of Africa. So how it is transmitted? So the infection is transmitted by eating infected crab or crawfish that is either raw, partially cooked, pickled, or salted. The larval stages of the parasite are released when the crab or crawfish is digested. They then migrate within the body, most often ending up in the lungs. So okay, for its pathogenesis, although the pathogenesis of human infection of P. westermani is not fully elucidated, experimental infections of cats or dogs could explain the early phase of paragonimiasis. As the larvae of P. westermani penetrate the intestinal wall and localize in the peritoneal cavity, there appears to be a considerable migration inside the abdominal cavity before they direct toward the chest cavity through the diaphragm. So approximately 20 days following experimental infection with metacercariae by oral route, first pathological changes can be detected in the pleural cavity with turbid or hemorrhagic exudation containing also numerous pus cells. So for its diagnosis, the infection is usually diagnosed by identification of Paragonimus eggs in sputum. The eggs are sometimes found in stool samples like cough-up eggs or swallows. A tissue biopsy is sometimes performed to look for eggs in a tissue specimen. And yes, this infection has also symptoms. So after 2 to 15 days, the initial signs and symptoms may be diarrhea and abdominal pain. These may be followed several days later by fever, chest pain, and fatigue. So the symptoms may also include a dry cough initially, which later often becomes productive with rusty-colored or blood-stained sputum on exertion. So how to prevent this kind of infection? So the best way to avoid this kind of infection is never eat raw freshwater crabs or crayfish. So we have to cook the crabs or crayfish at least 145 degrees Fahrenheit or negative 63 degrees Celsius. So travelers should be advised to avoid traditional meals containing undercooked freshwater across patients. So for the treatment, in most people, paragonimiasis can be cured with oral anti-parasite medications. Both praziquantel and triclabendazole are approved for treatment. So praziquantel is taken 3 times daily for 2 days, while this triclabendazole is taken twice a day or 12 hours apart. So okay for its references.